The overall goal of this protocol is to visualize phagosome formation as well as the precise site of phagosome schism in 3D in living macrophages using total internal reflection fluorescence, or TIRF, microscopy.
This method combines TIRF microscopy with epifluorescence to monitor the step-by-step localization of two different fluorescent proteins during pseudopod extension and tip fusion.
The technique provides a systematic method to determine the critical angle and obtain a TIRF signal, which is crucial for drawing conclusions about the events close to the plasma membrane.
The day before the TIRF microscope session, turn on the heating chamber to 37 degrees Celsius to allow a homogeneous heating of the microscope stage. The next morning, use 100 microliters of PBS supplemented with 0.1% BSA to wash 7 × 10 to the 6th sheep red blood cells, or SRBCs, per 35 mm glass bottom dish 2 times. After the second centrifugation, resuspend the cells in 500 microliters of rabbit IgG anti-SRBCs at a subagglutinating concentration in PBS supplemented with 0.1% BSA per 5 microliters of cells, and incubate the cells for 30 minutes with a slow rotation. At the end of the incubation, wash the SRBCs 2 times in PBS plus BSA.